Mitosis is the last stage of the cell cycle, lasting only an hour. Before beginning mitosis, a cell will undergo interphase in which the cell goes through 3 phases. In G1, the cell grows. In the S phase, the cell replicates its DNA, and in G2, the cell prepares for division. Prophase is the first stage of mitosis. In this stage, the chromatin fibers become tightly coiled and the nucleoli disappear. The mitotic spindle begins to form by the centrosomes moving away from each other due to the lengthening of microtubules. Centrosomes are the subcellular region at the poles of the cell that organize the microtubules. Microtubules provide structure to eukaryotic cells. The next stage is prometaphase. The nuclear envelope has fully disappeared by this point. After the nuclear envelope has disappeared, the microtubules will inhabit the area. The chromosomes become more condensed as the chromatids now have kinetochore. Kinetochore is a specialized protein structure where the microtubules attach. The kinetochore microtubules begin attaching to the kinetochore region of the chromosome, while the non-kinetochore microtubules interact with the microtubules that are on the opposite pole as them. Next is metaphase. The chromosomes will arrive at the metaphase plate in this phase. The metaphase plate is a plane that is equidistant from each pole of the cell. This allows all of the kinetochore microtubules to attach fully to the kinetochore of each pair of sister chromatids. The chromosomes then start to pull apart through a process of tug-of-war. Anaphase is the shortest phase of mitosis, lasting only a few minutes. Anaphase begins when the cohesion proteins are fully cleaved, allowing the sister chromatids to part and to go to opposite sides of the cell. They are now daughter chromosomes and fully fledged chromosomes. The microtubules shorten as they move the daughter chromosomes closer to the centromere region. The centromere region specializes DNA sequence to link sister chromatids. The cell grows in size as the microtubules lengthen in preparation for the cell to divide into 2. Telophase is the next step. During this phase, the 2 daughter cells form nuclei from the remaining fragments of the parent's nuclei. The mitotic spindles are depolymerized. At the end of this phase, genetically identical daughter cells are achieved. Cytokinesis is the final stage of mitosis. The cleavage furrow forms. The ring of acetin microfilaments interact with myosin molecules. The cytoplasmic side of the furrow is a ring of acetin. This causes the daughter cell membranes to cleave.